On the surface of your tongue, within structures called taste buds, are specialized cells. Each of these cells is studded with thousands of tiny molecular machines. For sweetness, the most important of these is a receptor made of two proteins joined together, T1R2 and T1R3. Think of it less like a simple button and more like a microscopic Venus flytrap waiting with its jaws open. When a sugar molecule, like glucose, drifts by and nestles into a specific pocket between these two proteins, the shape of the entire structure changes. The jaws snap shut. This physical movement is the very first step, the flick of a switch that begins the entire sensation we call sweet. But this molecular lock isn't perfectly secure. It can be picked. Consider a molecule of aspartame, an artificial sweetener. It has a completely different chemical structure than sugar. Yet, a part of it happens to be just the right shape to fit into that same critical pocket on the T1R2/T1R3 receptor. The same goes for sucralose, which is a modified sugar molecule, but one your body can't use for energy. To the cell, the identity of the key doesn't matter. All it knows is whether the lock has turned. The Venus flytrap snaps shut for aspartame just as it does for glucose. The cell sends the exact same message, sweet thing detected. Your tongue is being fooled, but it's a trick that starts at a single protein. That snap of the receptor closing triggers a silent, microscopic chain reaction inside the taste cell. It pokes a nearby G protein called gustacin, waking it up. This activated gustacin then sets off a cascade of other molecular signals, like a line of falling dominoes. This internal Rube Goldberg machine has one purpose: to open specific ion channels in the cell's membrane. When these channels open, positively charged ions flood into the cell, dramatically changing its electrical state. In that moment, a purely chemical event, a molecule binding to a protein, has been translated into the language of the nervous system, an electrical impulse. The message is being prepared for delivery. This electrical change races to the other end of the taste cell, where it triggers the release of neurotransmitters, primarily a molecule called ATP. These ATP molecules are dumped into a tiny gap, a synapse, between the taste cell and the end of a nerve fiber. This nerve is like a wire, but it doesn't just carry a simple sweet signal. The nerve endings here receive inputs from multiple taste cells, detecting bitter, umami, and sour tastes as well. The message sent to the brain isn't a single word, like sweet. It's a complex sentence, a pattern of activation across many different nerve fibers that the brain must then decode. The information is already becoming less about the molecule and more about the pattern. This patterned signal travels along cranial nerves towards the brainstem, arriving at a region called the nucleus of the solitary tract. This is the first major checkpoint, a relay station that gathers taste information. From here, the signal is forwarded to the thalamus. The thalamus acts as the brain's grand central station for sensory input, sorting signals and directing them to their final destinations. The taste signal is routed to a specific strip of brain tissue called the gustatory cortex, located deep within the folds of the insula. It is only here, in this final destination, that the raw electrical pattern is finally interpreted and you experience the conscious sensation of sweetness. But the gustatory cortex doesn't work alone. As you chew, aromatic molecules from the food travel up the back of your throat into your nasal cavity. This is retronasal olfaction, and your brain's olfactory cortex lights up, sending information about smell to be blended with the taste signal. This is why food tastes bland when you have a cold. The cherry flavor in a candy is almost entirely an aroma, not a taste. Furthermore, your brain's visual cortex chimes in. A red-colored drink is perceived as sweeter than the exact same uncolored drink. Your expectations and memories, housed in the frontal lobe, also preemptively tell the gustatory cortex what to expect. What you call taste is actually a multisensory hallucination assembled on the fly. So what are you actually tasting? An artificial sweetener proves that the sweet isn't inherent to sugar. It's a property of any molecule shaped correctly to activate a single protein. That activation creates an electrical pattern, which is then fundamentally reinterpreted based on what you see, what you smell, and what you remember. The sensation is not a direct perception of reality. It's a useful simulation, a story your brain tells itself based on heavily filtered and edited data from your tongue. The ghost in your mouth isn't the food. It's the complex, predictive model of the world your brain has built. You are not tasting the sugar. You are tasting your brain's idea of sugar.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.